Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the different methods of anesthesia and euthanasia in laboratory animals. Now let's start with the video. But first of all, let us know about what is anesthesia and what is euthanasia. Anesthesia. Anesthesia is the act of producing sensation-free relief from pain or pain-producing procedures. The statement says that it is an act or a procedure of producing sensation-free relief from the pain or pain-producing procedures, which means after the effect of anesthesia, the animal gets unconscious. The anesthesia must be performed by a person with knowledge of and familiarity with the drugs to be used in the animal species under consideration. The statement says that it should be performed by a well-trained person who has the knowledge of the drugs and is familiar with the drugs that are used in the anesthesia process. The commonly used anesthesia are, there are two types. Basically, first is the inhalational and second is the injectable. The examples of inhalational anesthesia are isoflurane, desflurane, and sevoflurane. And injectables are ketamine, diazepam, and xylazine. Now, what is euthanasia? Euthanasia means the humane killing or sacrifice of an animal which produces rapid unconsciousness and subsequent death without or minimal pain or distress to the animal. The statement says that it is the killing of the animal without feeling pain or giving minimum pain or distress to the animal. The choice of the method depends on species, age, availability of restraint, and the skill of the individuals performing euthanasia. There are two types of euthanasia methods. The first is the physical and the second is the chemical method. Now let's learn in depth about these methods. As we can see, here are the different methods of the anesthesia and euthanasia. Now let's start with the first anesthesia method. Here we have the inhalational and the injection. So now let's start with the inhalational method. In the inhalational method, first of all, we need a cotton swab, our anesthetic drug, our rat, and a closed chamber. The steps are that first we have to put the chemical on the cotton swab, our anesthetic drug, and then we have to place the animal near it and then enclose it with the closer. Now, as we can see in the video, the rat is getting unconscious with the effect of the anesthesia. The volatileness of the anesthetic drug is released and vaporizes in the air, and the closed container entraps it and creates a charged environment inside of the container. The drug is inhaled by the rat and it gets unconscious. The common agents are isoflurane, which is of 1 to 3%, sevoflurane, which is of 2 to 4%, delivered using a vaporizer and an oxygen flow, and provides rapid induction and recovery. This statement means this technique of the anesthesia provides the rapid induction of the anesthetic drug, which is inhaled by the rat, and it shows its therapeutic effect. And the recovery of the animal is also very rapid in this method. Now let's see the second method, which is of injection. In this injection method, first we need an injection which is loaded with the anesthetic drug, and then gently we have to hold our laboratory animal, which is our rat, and then we have to inject the anesthetic drug with the syringe in the intraperitoneal region of the animal. The common agents are ketamine, xylazine. Ketamine and xylazine are used in combination, and then we have pentobarbital and urethane. The site of the administration of the drug in this injectable anesthesia are intraperitoneal region IP or the subcutaneous region SC. It is suitable for longer procedures requiring deep anesthesia. This statement means this step of the anesthesia, the injectable anesthesia, is required when there is requirement of the longer unconsciousness of the animal. So it gives a deep and longer procedure and it is provided from the deep anesthesia dose. Now let's see the euthanasia process. As we know, there are two types of euthanasia process. First is the chemical method and second is the physical method. Let's see the chemical method of euthanasia. At first, we have the inhalational method. The inhalational method of euthanasia is like, first we need a container inside of which the animal is placed, And then we need a gaseous cylinder which provides the euthanation gas, which is used to euthanize the animal. As we can see here, it contains the euthanizing gas, which is filled inside the closed chamber where the animal is kept. The description of this method says that the carbon dioxide inhalational is a widely used method for euthanizing small laboratory animals such as rats and mice. The step says that for euthanizing, we use CO2 gas, which is widely used and which is very common in use, and which is good for euthanizing small laboratory animals such as rats or mice. The procedures are first we need a chamber. As we can see, this is our chamber. Animals are placed in a chamber where CO2 is gradually introduced. As we can see from the cylinder, we are introducing the CO2 gas inside the chamber and then the flow rate. The flow rate should displace 10 to 30% of the chamber volume per minute to ensure a humane and painless process. The statement says that the flow rate of the gas should be of the 10 to 30% of the closed chamber per unit volume. It means we have to introduce 10 to 30% of the chamber's volume inside the chamber so that a humane and painless killing of the animal is done. The advantage of this step is that the CO2 is effective, readily available, and is inexpensive. It is suitable for euthanizing multiple animals simultaneously. The considerations are a proper flow rate and a gradual introduction are crucial to minimize distress. Animals should be monitored to ensure death before removal from the chamber. The statement says that the proper flow of the gas and as gradually introduction inside the chamber is very crucial to minimize the distress of the animal. And the animal should be monitored before being removed from the chamber so that it means we have to monitor the animal's reflexes or the heart rate to ensure the death of the animal before removing it from the chamber. Now let's see the injection method of the euthanasia. This method is similar to the anesthesia injectable method, but the difference is that in the anesthesia method, we are using anesthetic drugs, which is used to unconscious the animal. But in the euthanasia process, we use euthanasia drug to kill the animal. The drugs that we use are pentobarbital, which is of 100 to 200 milligram per kg, which is given from the intraperitoneal or intravenous region. It is a commonly used barbiturate that induces deep anesthesia followed by cardiac and respiratory arrest. The pentobarbital drug induces deep anesthesia effect, which is followed by the cardiac arrest and the respiratory arrest. Now, second, we use ketamine, which is of 200 mg per kg, with the combination of xylazine, which is of 40 mg per kg, and it is given through the intraperitoneal region and can be used in overdose to ensure a painless euthanasia process. Then we use urethane, which is of 1.2 to 1.5 gram per kilogram through the intraperitoneal region and is sometimes used for terminal procedures but is less preferred due to prolonged effects. We use the intravenous injection, which provides the fastest action, while the intraperitoneal injection is commonly used but has a slower onset of action. Proper monitoring and confirmation of death, absence of heartbeat and reflexes are necessary before disposal of the animal. This statement says that the proper monitoring and the confirmation of death, it means we have to confirm the death of the rat by observing the absence of heartbeat and the absence of any reflexes shown by the rat before being disposal of the animal. Now let's see the physical method of the euthanasia. It contains three processes. The first is cervical dislocation, the second is the decapitation, and the third is the exsanguination. Now let's see the cervical dislocation. The cervical dislocation step is an easy step for euthanasia process, but we have to perform it very delicately because in this step we hold the animal from the head and then from the other hand we hold the tip of the tail and then slowly and in a sudden form we pull the tail so that the cervical bone break the rats and it instantly kills the rat. The procedure of this process says that cervical dislocation involves manual dislocating the cervical vertebrae from the skull to cause rapid death. The statement says that cervical dislocation, which means the dislocation of the bones of the cervical region, involves manual dislocating of the cervical vertebrae, which means it dislocates the cervical vertebrae from the skull to cause the rapid death of the animal. The application of this step is that the suitable for rodents weighing less than 200 gram of such as mice and less than 300 gram which is of rat when performed by trained person. Method. The head is held firmly while applying force to separate the spinal cord from the brainstem, leading to immediate respiratory and cardiac arrest. The statement says that we have to hold the head firmly while applying force to separate the spinal cord from the brainstem. The brainstem and the spinal cords are separated, which leads to immediate respiratory and cardiac arrest of the animal. The advantage of this step is that it is quick and it does not require any chemical agents and provides a clean blood sample for biochemical process. It is the main advantage of this step is that it provides a clean blood sample so that we can perform any further biochemical studies of the blood. The ethical considerations are that it must be performed only by a trained personnel following institutional guidelines, ensuring proper confirmation of the death. Now let's see the second physical method, which is the decapitation. In this method, with the help of surgical scissors, we directly cut off the head of the animal and within an instant the animal gets killed. It is a very painless method. And the procedure of this method is that decapitation involves the complete severing of the head using a guillotine or a sharp surgical scissors. The application of this step is commonly used for studies requiring uncontaminated blood or brain tissue, especially in the neurochemical research. This step also gives us the uncontaminated blood samples or the brain tissues. The method is that the animal is restrained and a sharp blade is used to ensure a quick and humane procedure with minimal suffering. This step gives the minimal suffering to the animal within an instant. The advantage of this method is that it provides a rapid, chemical-free euthanasia method without affecting blood composition or brain activity. And the ethical considerations are that it must be performed by a well-trained personnel by institutional guidelines and pre-anesthesia is recommended for larger rodents. We have to provide pre-anesthesia so that the animal does not move or disrupts the process of euthanasia. Now let's see the last step of the euthanasia process is that the exsanguination. In this step, we hold the animal gently and tilt it so that the abdominal region is visible. And with the help of a forceps, we make a cut and a deep cut in the vital organs of the animal so that a large volume of blood is drained and the animal gets killed instantly. The procedure says that the exsanguination involves the removal of a large volume of blood leading to hypovolemic shock and death, which means this step, due to the drain of the large volume of blood, it leads to the hypovolemic shock to the animal and causes death of the animal. The application of this step is that it is used in studies requiring a large blood samples for biochemical and hematological analysis. And the method is that it is performed via cardiac puncture or major blood vessels, example posterior vena cava, under deep anesthesia. This statement says that the method of this step is that it is performed when we put a cardiac puncture or we damage a major blood vessel so that a large volume of blood is drained. Example of the major blood vessel, we can say that it is the posterior vena cava, which is located in the upper abdominal region, and this step is done under the deep effect of the anesthesia. The advantage of this step is that it provides an uncontaminated blood from the uncontaminated large volume of blood and the researchers while ensuring a humane death when combined with anesthesia. The ethical considerations are that it must be always performed as a terminal procedure under anesthesia to prevent pain and distress. Now we have come to an end of this video. We have completed our experiment. I hope you have liked the video and thank you for watching this video. And for more such learning content, follow and subscribe our YouTube channel and follow us on our LinkedIn profile, on Instagram, as well as in Facebook. We update our daily posts and videos over these platforms. Thank you.